My name is Jaehnig. I'm a scientist in the lab with Dr. Bing Zhang at the Baylor College of Medicine. My poster is entitled Accurate Inference of Kinase Activity Connects Genetic Aberrations to Signaling in Human Tumors. CPTAC has profiled multi-omics data for over 1,000 tumors from 10 different cancer types. The types of data that they have collected include whole exome sequencing and RNA-seq data, mass spec-based proteomics data, and mass spec-based phosphoproteomics data. Here we focus on utilizing phosphoproteomics data from 8 of those 10 cancer types to assess different methods for inferring kinase activity in a given tumor by using measurements for known kinase substrate phosphorylation sites. The methods that we will be evaluating for inferring kinase activity from phosphoproteomics data generally fall into 2 categories. Methods where scores represent aggregate measurements of all known targets in a given sample, or methods where scores come from comparison of all target sites to other sites in that sample. Figure 1 outlines our strategy for benchmarking these different methods for inferring kinase activity. We first curated a list of activating sites on kinases from PhosphoSite Plus and manual review of the literature. Using these sites, we identified a set of gold standard kinase-tumor pairs from each CPTAC phosphoproteomics data set. An example for how we selected kinase-tumor pairs to include in the gold standard set for benchmarking kinase activity is shown in this histogram of an activation site on map 2K4. Tumors in the top 5% of the normal distribution for the site were selected as map 2K4 kinase tumor pairs for the positive set because they are the tumors in which map 2K4 is most likely to be activated, whereas tumors in the bottom 5% are those in which map 2K4 is most likely least active and were thus selected for the gold standard negative set. The table on the right shows the numbers of kinase tumor pairs in the gold standard sets when this selection process was applied to each cancer type. For ROC analysis, we first converted the activity scores to z-scores within each data set, then evaluated how well those z-scores discriminate between the gold standard positive and negative pairs for all cancer types combined. Figure 2 shows the results from ROC analysis of kinase activity scores inferred using 8 different methods. The box plots show the median and entire quartile ranges for AUROCs from 1,000 random samples using 80% of the gold standard pairs. A simple score based on the mean of all target sites performed the best, while KSEA, the target site median, and the Wilcoxon test were all very close behind. The mean AUROC values for each method are also shown in Table 1. In addition, the table shows the results when the analysis was repeated with a less stringent threshold for selecting pairs for the gold standard set. Top and bottom 20% versus 5%. And the average correlation between kinase activity scores and the activating sites chosen for establishing the gold standard set for the corresponding kinases. Similar trends are observed for all metrics, with the mean score showing the best performance. Since the overlap of known targets with sites measured in global phosphoproteomics datasets is usually fairly poor, we next tested the hypothesis that kinase activity inference could be improved if we include predicted targets in addition to known targets for a given kinase. Figure 3 shows that this is indeed the case. Adding targets predicted by networking improved performance significantly for all methods tested, and the mean score of known plus predicted targets showed the highest performance, with KSEA using the combined target set being close behind. Next, we took advantage of matched CPTAC genomics data for the same samples in which we inferred kinase activity to identify cases where gain-of-function genetic aberrations result in altered kinase activity. The box plots in Figure 4 show that tumors with potential gain-of-function alterations in the EGFR gene showed higher EGFR activity than EGFR wild-type tumors, from glioblastoma and lung squamous and adenocarcinomas. Furthermore, as shown in the bar plot on the right, upregulated kinases in EGFR gain-of-function tumors showed enrichment in KEGG pathways downstream of EGFR, including PI3K, AKT, and RAS signaling. Since there is a greater need for finding therapeutic targets for cases where tumor suppressors are lost, we systematically applied this approach to identify potential kinase targets in tumors with genomic loss of function in tumor suppressor genes. The box plot on the left provides an example where loss of the CDKN2A tumor suppressor results in upregulation of a downstream kinase, CDK6, that is targetable, while the diagram on the right shows kinases from the cell cycle that are upregulated when TP53 is lost in breast cancer. Notably, evaluation of published CRISPR dependency data sets suggests that many of these kinases are potential therapeutic vulnerabilities in cell lines with loss of TP53. In summary, we utilized activation sites on kinases to establish a gold standard set of kinase-tumor pairs for high and low kinase activity to use as a benchmark for methods to infer kinase activity scores from phosphoproteomics data sets. While most methods showed similar performance, the mean of all target sites for a given kinase consistently performed the best. Furthermore, adding predicted targets from the network and significantly improved performance for all methods tested. Finally, CPTAC's multi-omics data facilitated the identification of potential therapeutic targets in tumors with genetic gain of function in oncogenes or loss of function in tumor suppressor genes.